So as we all know that data regarding the data, so as most of us working on the aquaculture, most of us aquaculture related work or maybe marine science, fisheries, aquaculture. So everyone is related to the stream or the field where we are collecting lot of data. And the thing is that the science of data analysis that is not related to only the aquaculture, not related to only fisheries, not related to marine science. Anywhere, whenever you want to go, you will find out data. So data is from coming from everywhere, whether it is academic sector, whether it is industrial sector, whether it is country or any sector. So telecom industry, any industrial sector, there is a lot of data. So what the means of this data? So at the initial stage where the data does not have any meaning, so it's just a number, it's just a number. So how we will make this number into a valid conclusion? So that is a process. Okay, so we have collecting the data, there is a lot of data, but until or unless we are not analyzing the data with proper tools, we are not interpreting the data with proper interpretation, we are not going to conclusions. Okay, so ultimate goal is to draw a solid or draw a valid conclusion that can help you to interpret your data to result to everything. So that's why we need a data analysis techniques. So that will there is a lot of data analysis techniques, but which analysis will be applied? So this is also very important. We'll talk about little bit of selection of analysis. So the thing is that I already talked about it. Whatever is the career it is related to academics, related to industrial, anywhere. So there is a lot of data, and to make a professional decision, we need to analyze the data to make a valid conclusion. So that's why the importance of data analysis is very essential. It is not only one sector. So data analysis is coming from every sector. And as a student, as I might be wrong, but as a student of aquaculture or the fisheries or biology, means we are basically coming from not related to data analysis background. So we are coming from a biology, more mainly biology or aquatic science background. So there is a we are afraid of the kind of mathematical or data analysis techniques. Most of it's not for everyone, but yes. So that's why that there is a lot of data we are collecting every day for our from our field, from our lab, from our experiment. But after that, what we will do with that data, that we cannot able to understand. Okay, or we have to go to a statistician, we have to go take help with someone who is able to help me to do the test for me, t-test or ANOVA, what will go for a regression or correlation. So that is the major problem for the that science of related to biology, or maybe that not related to the data science. So first of all, what is the difference between the data, information, and statistics? So that three are three different things. So data is only the number digit. Data is nothing, only the number or digit. Okay, and what is the information? So from the data you are collecting the informations. Okay, so just from these tables you can find out that here is a data of 20 individual and their body weight. So here is that data. If you go to the only data, you cannot find out anything. But if you try to extract the information from the data, you can find out, yes, from 5 individual of this group is from belongs to this range, 15 individual of this group is belongs to this range. So this is kind of information I'm extracting from my data. But this is not the statistics, this is not the interpretation. So after that you can make a statistics, you will find out a mean weight of this group is this one, and median weight of the whole group is this one. So then you can find out something, a statistical analysis, then you can interpret your results. Now comes to the data. It's consists of coming from different observations, it's coming from counting, it coming from measurement, it coming from different response. So we are always uh, counting something, we are measuring something, we are observing something, then we are collecting the data. And so then after that we are analyzing the data through data analysis tools, and that is called the statistics. That is the science of collecting, organizing, and analyzing and interpreting the data to reach a valid conclusion. So that is the statistics. So before going to that data, we have to little bit learn about the what is population and sample. So populations is the whole things. Okay, so suppose you are a agriculture scientist, or a aquaculture scientist, or whatever it is, or a fishery scientist. You are not going to catch all the tuna from the ocean. You are not going to measure all the tuna from the seas. It is not possible. Nobody cannot do these things. Okay, so that is the tuna population in a particular area. So what we will do, we will take some samples from the populations, then we measure it. Okay, then we put the interpretation, yes, this is the standard mean of that populations. Okay, so that is the difference between the sample and population. So sample is the subset of the population, because the whole population cannot be measurable. Yes, it is time taking, but it is not. We are usually basically collecting the sample from the population and we are measuring the sample. So that is the difference between the sample and population. Now I have already talked about it. Whatever sector you will go, whether it's education sector, manufacturing sector, we know about the epidemiology, or we know about the last COVID. So whatever the data, every day COVID data is coming, and it's represent a way that people can understand how much percentage or whatever it is. We can check, we can see the COVID statistics. So governmental sector, population statistics, whatever it is, the income, national GDP, everything. So all are calculated, analyzed, and then government should take the decision on based on the interpretation, or based on the analysis of the data. Environmental sector, aviation sector. So data is coming from everywhere. So data analysis, it's a common branch, or I can call it's a common branch for all the sector of our requirement. So we have already talked about it. It is basically organizing, representing, analyzing, and to reach a decision. So now comes to the type of statistics or type of data analysis. There is a, we can find out the descriptive statistics and inferential statistics. So descriptive statistics, it basically tells about the structure of the data. It basically tells about your summarizing of your data. Some you are getting some information out of it. So descriptive statistics, if you go and only do the descriptive statistics, you can find out a lot of very important informations, just like starting from mean, median, mode, standard deviation, standard error, kurtosis, skewness, confidence interval. So different things we are getting. So what does it means that kurtosis, skewness, confidence interval, mean, median, mode? We most of us we know about it. Mean, median, mode, standard deviation, standard error. So that's basically representation of data analysis everybody did. And inferential statistics is that is used to, after collection of the data, you have to do particular test, you have to do some determinations, and then there is, we have already talked about the populations and samples. Then we have to take some samples from the population, then we will do some statistical analysis. Inferential statistic means we can wants to find out the relationship, we can find out the whether that there is any variations or any relationship, this kind of inferential statistics. So inferential statistics, what are there, we will go here. So descriptive statistics is also different part: univariate, bivariate, and multivariate. As we all know, uni means one sample, bivariate means two sample, and multivariate means multiple sample. And inferential statistics, when we are going to apply the data to form based on the mean. So basically how much it's the this both the variables are significantly different from their mean. Okay, so there is this inferential statistic basically apply to the mean of the data. And so univariate, we know the shape of the data. So kurtosis and skewness, it basically tells you the shape of the data, or whether it's a normally distributed data, Bell's effect curve or Hodge kind of curve, you will find out. So we know about the normally distributed data, non-normal data, we'll talk about it later on also. So shape of the data, this is very important. First of all, where this shape of the data will help you whether you go for a normal means a parametric test or non-parametric test. We'll talk about it later on. So also, but shape of the data is very very important. Okay, sometimes the data is skewed data. Okay, so then we have to think about a different test. So that is very important. That's why we are, most of us, we basically means we know the test t test and but whether we will do the t test, whether we will do the Kruskal-Wallis test, or whether we will do the different test, that is very important. Then center tendency, mean, median, mode, these are basically tells you the center of the tendency of the data, center of the data. Then spread, how much the data is spread. Okay, so that's confidence intervals. Okay, or that will tells you that how much data is spread, or mean or minimum or interval. So that will tells you that spreading of the data, and the relative position, where is the position of the data. Now comes to the bivariate. You have, when you have two variables, then you can do one thing, that you can do find out the relationship between these two variables, and you can, from this analysis, you can also predict. So if you want to find out the relationship between the two variables, that you have to do the correlation. And when you want to find out the prediction, means one variable is dependent to another variable, then that time you have to go for a regression. Okay, and multivariate, multiple regression, multiple correlations is also there. So it depends upon the how many variables you have. If you have a multiple, more than two is called multiple variations. Now apply to mean, you have two group. If you have two group, then you can do the t-test. If you have more than more than two group or more than two variables, then you have to go for a ANOVA test. Okay, so that are the very very simple deviations of your different statistical test. Now central tendency, we all know mean, median, mode. Yes, we know, very simple thing, nothing mean. But important thing is that that is basically is also one of the most important, that from the mean or median you can find out which sample you will go for calculate the mean, which sample you are going for calculate the median. Okay, so that is also depend upon the shape of the data. If the data is skewed data, then you can go for the median, not mean. Okay, if data is normally distributed data, then you can go for the mean. For the mean of the data is very very appropriate. But if you cannot calculate median for a normal data, normally distributed data, or you, that will not be statistically valid. So that is very important. The shape of the data and mean, median, mode will be three different central tendency, central measurement, where are the means your data, center of the data is where it varies. Okay, where it stand. So that central tendency. And then variability measurement. Okay, I know I already talked about that, how much data is varies or how much data is spread. So variability, it's basically tell about the interquartile range. You, if you go for a any what is called means range, mean to max, or any variation. So standard variation, standard deviations, and coefficient, or I can call the confidence interval. So these are basically tells about the variability measurement. Okay, and that interquartile range is also very important. If you want to read a graph from the data distributions, then you can find out that how the interquartile, so 25%, 50%, 75%, 95%. So the distribution of the data is where. Okay, so the from median or mean or interquartile, we can, so first quartile, second quartile, third quartile. So 25%, 50%, or 75% confidence interval. So that does it mean that just like in confidence intervals. Okay, we are talking about 95% confidence intervals. So what does it mean? So it means if you take any sample from that populations. Okay, after measuring, I have got, after measuring of 50 or 100 tuna length, okay, or any fish length. Okay, I find out the confidence interval is varies from this value to this value. Okay, confidence interval varies, suppose I am taking to 21 to 31. So what does it mean, that confidence intervals? Okay, 95% confidence. So that means if Now after doing this sampling, so if you go to that same populations and if you measured or take any sample from that particular population, and if you measured again 95% time, you confidence that the mean length or the length will be varies from this value to this value. So that is mean the 95% confidence interval of your data. That then the variance, how much the data is varies. Standard deviations, we all know standard deviation means it's from the mean, how the data is varies from the mean. Okay, so if you have the samples of 4 5 8 9, so you have find out the mean is the 5, so then how is the 2 is varies from 5, 4 is varies from 5, so that is called standard deviations. Okay, and standard error is also find out the how much accurate is your data. Okay, so it will tell you about the accuracy of your data. For it's basically standard error, it's basically tells you that yes, how much accurate is your data, and the mean is likely to be. And skewness and kurtosis, this is two very important parameter that can be measured, and from these values of skewness and kurtosis you can find out the shape of the data. So positive skewed data, some of the time that you are taking all the data related very high frequency or something in particular. So in I can from suppose a piece length. Okay, piece length, suppose you are getting a highly positive skewed data. What does it mean? What does it mean? So positive skewed data, so if this one figures is tell you starting from lowest range to highest range from this data, what is that? So it tells you that in your sample or in your populations of these particular species of fish, very highly young individuals are very much dominating in these populations. Okay, because the length of this lower individual, low size or the short size or the young individuals are very high in the population. So yes, so distributions is very important. And the zero skewed, basically that means the normally distributed data, there is no skewed data, so it's normally distributed data. And also in the negative skewed data you can see that suppose if I take the length of the piece, then from this negative skewed data you can find out that you can tell that this population is dominating by the larger piece. Okay, if I take the length frequency data. Now kurtosis, this is skewness, and kurtosis is also basically tells you that height of the peak. Okay, so if there is a something, one data is going out of outliner maybe, or there is a peak of the data. Okay, something, so suppose COVID. Okay, so it's going normally, then some after certain period it's have a particular peak. Okay, so that height of the tenure of data distributions, it's basically measured by the kurtosis. Now comes to the different between the difference and relationship. So difference and relationship, it's very important. So if you want to find out the difference between your variable, okay, so if you have two varieties of banana, suppose, okay, so if you have a one variety 1 and variety 2, so now you want to check the which variety is much more productive. So which test you have to do here? So because you are find out the differences between these two variety. So I told you that t-test is used for the only two variable. More than two variable, you have to go for ANOVA. So if you are measuring the banana production between the two variety, then you have to do the t-test. But in between the t-test, there is also different type of t-test: paired t-test, unpaired t-test, one tail t-test, two tail t-test. Okay, so different type of t-test is also there. We will can discuss later on also. And ANOVA, whenever you are measuring the more than two variables. Okay, so that's three. Suppose you are measuring that production of banana. Is there any significant variation of the banana difference in the banana production in the three variety: variety 1, variety 2, and variety 3? At that time, you will do an ANOVA test. Okay. Now comes to the relationship between the variables. So if you want to find out the relationship between the two variable x and y or whatever it is, then you have to do the correlation or regression. Suppose one example, that temperature and ice cream sale. Okay, so if the temperature increase, the ice cream sale will be increased. So summertime too much ice cream sale, winter time ice cream sale is reduced. Okay, so that is, so you can find out the correlations between the temperature sale and ice cream sale, temperature increase and ice cream sale. Yes, there is a positive relationship, positive correlationship between temperature and the ice cream sale, and this is correlation. But what is regression? Okay, I have a data for, suppose one ice cream shop. I have a series of data. Okay, particular temperature, ice cream sale, temperature, ice cream sale, temperature, ice cream sale. Okay, I have this series of data. Now I want to plot this data into a regression model. Then after that, I want find out the regression model. That is, we all know that simple equations of regression, that y is equal to mx plus c. Okay, so linear regression. So ice cream sale and temperature. Now we plot this data and we have a fit the regression line. We have got uh, the slope and intercept, and from that one you can predict. So yes, I have this data. I have did the calculation. I have run the regression model. Now, if I tell you that temperature is 32 degree, from this regression model you can predict, you can, at prediction it is not accurate, 100% not accurate, but it can be, you can predict that yes, 32-degree temperature means the ice cream sale will be this much, 34-degree temperature means ice cream sale could be this much. So that is called regression. So prediction analysis can be done from the regression analysis. Now comes to the association. Already talk about it, correlation. So this is the association between the two variable. So correlation basically varies from 0 to 1, and correlation values. And so we all know that 1 means highly correlated, and 0 or close to 0.5 means it is medium correlated, or close to 0 means there is a no relationship between the variable. Okay, so so this is the simple correlation. So relationship between the two variable, and different r values will tells you the different relations, how much relationship is strong. Okay, between the variable x and y. And regressions, that is basically the association between the two variable. Okay, I told you that x and y and how much they are associated. Yes, if ice cream sale, I can predict the ice cream sale, okay, with given the particular temperature. Okay, so that is the regression. So look the significant relationship between the two variable, and another one is also predict the one variable for given values of the other. Okay, so if I give you the value of temperature, you can give me the value of ice cream sale. Okay, you can calculate, you are able to calculate the ice cream sale. So that is the regressions. Now comes to the non-parametric test. Yes, it is very important, because it's depend upon the shape of the data, it depend upon the normality test, different things. So basically what we will do first, whenever any test, before running any test, first whenever you have the data, you have to do a normality test. Okay, there is a different normality test, superior test, different normality test there. Even if you from the shape, from the data distribution, you can find out the distribution of the data, then you can find out if the normality test, the data is non-normal, then you have to apply non-parametric test. Okay, you cannot do a non-normal data t test. Okay, so for which condition you will do a non-parametric test? First of all, if the data is ordinal, okay, data is not nominal, so data is ordinal. So data is coming from something just a ordinal orderly data, or something, sometime the questionnaire survey, or it's a I can say sometime not a quantitative data, qualitative data. Okay, so qualitative data or assumptions, and then very skewed data, that is very, I told you several times, that's very skewed data. Highly skewed data means it is not normally distributed, and also very small sample size, that, and very highly influential of outer line. What is outer line? We will say yes, if you have a data something, so if you feed the data, then you will find out the some of the data points are going out of the range. Okay, so they are called outer line. So if you have a very high outer line, so that time also you have to talk about, you have to do the non-parametric test. So which kind of non-parametric tests are there? So instead of t-test, if you want to do a independent t-test, so instead of t-test, you have to do mean Whitney test. Instead of paired t-test, you have to do the Wilcoxon rank test. Instead of ANOVA, you have to do Kruskal-Wallis test. Instead of repeated measure ANOVA, you have to do Friedman test. So Pearson correlation, you have to do Sparkman correlation. Linear regression, you have to do non-transfer of the data. So these are the different format of parametric and non-parametric test. This is very important. So if you have the data, first do the normality test. Okay, whether your data is normal or non-normal. If it is normal, then you can do the normality parametric test. If data is not normal, then you can go for a non-parametric test. Normality test, I already talked about it. So if you have to check the shape of the data, there is a lot of tests are there. Sapiro and the different other test is there. You can do normal test and you can so check the shape of the data. Then you can go for parametric and non-parametric test. So these are the some of the test type. Now I'll talk about little bit about the data collection and hypothesis. Okay, so that is also very important for the data analysis, hypothesis testing. So data collection technique, that is very easy and very simple way I can represent here. So if you have using available information, so that is the secondary data, available information, something checklist, governmental report. Okay, so different where already available data. So data collection tool is basically data compilation, different secondary data you can use. Now observing and measuring. So if you observing how many fish are there, you are measuring the fish length regularly. So this can be data collection tools. This is this interviewing. Some of the time we are using social studies of it related to aquaculture, even in fisheries. We will do sometimes social studies or socio-economic of fisheries or aquaculture. We will have to do some interviewing data, and we can use the 1 to 5 Likert scale data. We can do questionnaire survey, we can interview the farmer. So this kind of data also can be used for interviewing, and some of the questionnaire also, some of the administrative question, because research that does not mean only doing the research only getting publication is not research. Is until the research will does not make any policy making, it does not make any much more sense. So ultimately you have to go to the administrations or make a policy. So at that is also a questionnaire survey that can be used for that particular. So sampling methods, yes, or you all know about the sampling method. So different sampling method here I already only describe four. There is a lots of sampling methods are there. Simple random sampling, as we all know, we are collecting the sample from here, there. So randomly I'm collecting the sample. Systematic sampling, yes, I am doing sampling every month, first week of every month. Okay, first week of every month. That is my systematic sampling. Okay, so I am doing sampling of particular time period of a month, particular time period of a year. So systematically I am collecting the sample. Stratified sampling, yes, I am collecting the sample from different strata. So different strata means, first of all I have to, there is a populations. Okay, so I'm collecting the sample from the population, then I will classify, I will make the strata into all the samples into different strata. So this is large, this is big, this is medium, kind of this strata. Then I am taking something, some of the sample from large, some of the sample from medium, some of the sample from the low or small. So that is called stratified sampling. Multi-stage sampling, it is very easy. Multi-stage, it is by word you can understand. The multi-stage means it have a stage. So you are collecting the sample from starting from suppose a village level to country level. So that is called multi-stage. So I'm collecting this data from the village level, I'm collecting the data, then the district level, then the state level, then the country level. So the multi-stage, I'm collecting the sample. So that is called multi-stage sampling. So data representation, as we all know, so graphical way, so nothing to talk about it, but it should be a separate training program on that data represented, different graph type, we all know about it. So bar, pie, scatter, linear histogram, infogram. So where you will apply this particular graph, that is also important. Okay, you have something short data, you cannot apply a line graph or a histogram or infograph for this particular short series of data. So that is also. Now comes to the hypothesis testing. It is very interesting. So first, one slide I'll describe the hypothesis testing. I try to. Okay. So first of all, whenever you are going for a particular research or any research, first of all you have to identify your research problem, or that is called research question. You have to identify your research question. So whenever you identify your hypothesis or your research questions, then after that you can go for a research. So first of all, you have to, each and every research must have a hypothesis. Each and every research must have a, without hypothesis, to me none research cannot be done without hypothesis. Okay, so first of all a research question should become. So what is the, here I am describe with one example with hilsa. And hilsa is one of the fish, migratory fish in Bay of Bengal or in Arabian Sea also. So that basically comes to the freshwater for migration, and during, if the salinity will be reduced, when the freshwater too much freshwater input, it comes to the river for migration. Okay, so this is the hilsa. So that is the basic we know. So we want to, in this research question, so what we will do, so we want to find out, is there any relationship between the hilsa catch and seawater salinity. So this is my research question. This is my I can say this is my research question. Okay, so is there any relationship between hilsa catch and seawater salinity? So what is my hypothesis? I'm taking one hypothesis. Yes, might be I am taking one hypothesis that yes, there is a relationship between the hilsa catch and seawater salinity. Suppose, so I am a positive, I am pessimistic, so positive, so I am taking yes, there is a relationship. Now after we have to, what we have to do, so this is my done, this is my research question. So what we have to do, we have to collect the data on hilsa catch. Also we have to collect the data on seawater salinity. Okay, then we have to do a test. Then we will to find out, we have to establish my hypothesis, whether I have take my hypothesis is correct or wrong. If it is right, then we have to go for null hypothesis, alternative hypothesis testing. Okay, so now we are going to field and we are collecting the data on hilsa catch, time series of hilsa catch, 10-year, 15-year, 20-year, whatever it is, and we are also collecting the seawater salinity, maybe from different data source, maybe satellite or maybe uh, regular sampling, whatever it is, we can collecting that this both the data. Now we will do a analysis. Which kind of analysis we will do? We will find out the relation. So relationship means we have to do a correlation. Okay, so here is no t-test, no ANOVA, because t-test and ANOVA is comparison between the variable. Relationship, when it is comes to relationship, either we have to do a correlation and regression. So correlation we will do first. So we will find out that there is a relation between hilsa catch and salinity or not. And if there is a very good relationship and we can do a regression model, we can find out a yes. So then we can predict the hilsa catch with given particular salinity. But those all things are depend on the statistical result and different things. Okay, is there also. So I'm not going to into the deep. So then we will do a correlation test. We'll find out the r square is good. We'll find out the probability p value is also significant. Then if you get a probability value is less than 0.05, means you are 95% confidence that your means relationship is okay. So at that time what you will do, you have to take a decision now. Okay, so now it comes to your interpretation and conclusion, your result. Okay, you got your result, you have to come to your interpretation. What your interpretation? Now you have to reject your null hypothesis. Why? Because that stress is statistically significant. Okay, so it's statistically significant. So my decision is the, I have to reject my null hypothesis. So if I so I have to write down my hypothesis. Null hypothesis is there is no relationship between the hilsa catch and salinity, and alternative hypothesis there is a relationship between hilsa catch and salinity. So this is my decision, my interpretation. Yes, there is a strong relationship between hilsa catch and seawater salinity, and hilsa catch increase due to decrease in seawater salinity. Okay, so this is a one slide I can talk about from research hypothesis testing. Now we almost all know about the null hypothesis and alternative hypothesis. So basically we are unable to reject the null or accept the null. Okay, we cannot talk about the alternative hypothesis. So whether you have uh, so whether you have going for a statistical any software or any understanding or any analysis, then you have to find out that you whether you reject the null or you are accept the null. Okay, so based on that one you will take your decision. So is there association or no association? If your probability is less than 0.05, that time you have to reject your null hypothesis, means you have to accept your alternative hypothesis. Alternative hypothesis is always is saying that there is a association, and if your probability value is less than 0.0 greater than 0.05, that you are not able to reject the null, so you have to accept the null. So that means there is no association. Now comes to the choose the right test. Okay, which test I will do? I will do the t-test, I will do the ANOVA or regression or correlation or one way ANOVA, two way ANOVA, one sample t-test, two sample t-test, parametric, non-parametric, Kuskil wale. So but this is very confusing. So which test I will do? This is very very important. So there are several steps to decide it which test you will do. So when you are going for a research project, first I already discussed, you must have a research questions. When you have a research questions, then you will find out your variables, and that variables you have to identify your dependent variable, independent variables. This is very important. And how many dependent variable is there, and type of the type of the data. So independent and dependent variable, that is very very important. That particular variable is dependent on the another variable, so whether it is independent or dependent variable. So that is, and then you have to find out, is there any relationship or is there any association relationship, or is there any variations. So I want to find out the variation between the two variable, then you have to go for a t-test. If you have more than two samples, then you have to go for a ANOVA, that we have already discussed. And if you want to find out the relationship, then you have to go for a correlation and regression. If you go for a prediction, then you have to go for a regression model. So this is very simple way to I'm not going to deep into the statistics, because this is very simple or basic statistics. Yes, clear research question is very important, and which variables help to answer the questions, it is also important. Just think about the example of Hilsa catch and seawater salinity. Now here we already discussed this thing. This is graphical representation. So two between the two groups, so you have to do independently test or more than two, one way ANOVA measured within the same subject, paired t-test. So this is also paired and non-paired t-test or paired t-test, and repeated measure ANOVA. Now if you have association or dependent variable, independent variable, so association or relation, correlation for two and more than two, multiple correlation. Even the correlation also have Pearson, Spearman, high correlation, couple correlation, because if your variables are continuous or normally distributed, then you can go for a Pearson correlation. Your relationship between the two variable are not continuous, it is a orderly or nominal, then you have to go for different correlation technique. Regression, linear and non-linear. Okay, sometimes we have apply for non-linear regressions and multiple regression. If you have a more than two, just a fish catch is dependent on the seawater temperature, salinity, nutrient, so different. So catch is dependent on salinity, temperature, nutrient, so multiple variables. So I have to do a multiple regression model. So these are the different into if you are going for a deep, then it can be. Now this is also very of the one of the important data type, that is first of the you have to whether it is scale data or categorical data. If it is a scale data, then two type of scale data, one is continuous and one is discrete. Continuous means it's a continuous series of time series or measuring the take of any value. So continuous value, and discrete means is count or integral, it have a limit, because the discrete means it is counting or not a continuous data. Ordinal data and nominal data. Ordinal data means it has a order, so it's have different order, or order 1, order 2, or stage 1, stage 2, stage 3. Okay, so this kind of ordinal data. And nominal data, it's age, sex, material status. So these are the nominal data. Or binomial data is two. So age is not a binomial data. Marital status is a yes or no. So that's the binomial data. Sex, male or female. So that's the binomial data. Age is not a binomial data. It's a normal nominal data. Age can be varies from different. But yes, these are the different type of data. This is also important to know. Now ultimate tools, yes, there is lot of statistical tools is available in the market. Yes, starting from Excel to SPSS, data, Statistica, different tools are there, and R, whatever it is. But most of the time we are rely on R because it's a free. First of all, it's a lot of resources available, and because other statistical tools that just I can say the SPSS or Statistica, you need a valid license. Okay, so these are the not free, so these are the paid and costly software. Okay, so R is very easy and free. It available for all kind of operating system, and it is a continuous process. Okay, so it's kind of I can say it's for it more or less it for a continuous improvement is there. It is not stoppable. It's a regular things are coming new in R. So R is the interface, and nowadays R Studio. R is the interface. R Studio is the working software we are using. R Studio for any kind of data analysis. So that's all. Yes, thank you for listening.